My name is Hunter Lanovoi. I'm a trainee in the neurosciences at Rutgers University. My mentor is Ioana Carcea. And today I'd like to talk to you about internal visceral states and social behavior. So you can see here is a study of over 18,000 adults of both sexes. And what they find is that social integration is inversely associated with the risk for chronic disorders such as stroke, asthma, heart disease, and bronchitis. What we can appreciate from this is that it's not clear if being social leads to better health or if those in better health have a propensity to be more social. Begin approaching this question, the anatomy and physiology of social brain networks in humans, in primates, and in rodents have begun to be defined. What you can see here is that a key node in this social decision-making network is the amygdala. This area also receives information from the body. And in regard to information from the body that's important for social behavior, this is a study in wild baboons in which social group membership, which is measured by the strength of the grooming relationship between the animals, and the composition of bacteria in the gut, termed the microbiome, was measured. What you can see is that microbiome composition is most similar in individuals that have similar social networks. You can see this on the right by this red group here. So the question then becomes, are these bodily signals from the microbiome necessary for social behavior? So evidence from this study suggests that this is indeed the case, that microbial signals are important for normal social behavior. So this is a study in rodents in which they raised mice conventionally with microbes, termed CC, or in a bubble devoid of any microbes, termed GF or germ-free. They then assessed the tendency of these mice to interact socially using a 3-chamber apparatus in which an interacting mouse has the choice to enter and interact with an empty chamber or interact with a chamber containing another mouse. What you can see is that in comparison to the conventionally housed mice, these germ-free mice show a significant decrease in their social interaction time. This behavior can be restored by reintroducing microbes. One hypothesis for why this relationship between the brain and gut microbes might have come about is that it might be necessary for preventing the spread of pathogens. And indeed, this is a study in which they use lipopolysaccharide, a component of the bacterial gram-negative cell wall, and also an inflammatory agent. What they find is that treatment with this component of the cell wall decreases the propensity for mice to interact socially in a dose-dependent manner. So what brain areas both respond to internal cues from the body and regulate social approach and avoidance? What you can see here is a brain network of cortical and subcortical regions that respond to social and internal cues and also regulate approach and avoidance behavior. The amygdala, which is one node in this network, both receives bodily cues and also expresses the receptor for the prosocial oxytocin peptide. So the parabrachial nucleus, denoted here by PBN, is an area of the brainstem involved in the relay of information from the body to the amygdala. And one important cell population in this region are neurons that express CGRP. These neurons have also been shown to respond to threatening internal stimuli, such as an inflammatory state. In this area, there is also a population of OXTR expressing neurons. These neurons are closely situated to the CGRP population, and they were found to be responsive to non-nutritive water intake. However, it's still unclear how this cell population of OXTR neurons might respond to other bodily stimuli, such as those from gut microbes, or how they might contribute to social approach and avoidance. Therefore, in our lab, to first define this putative circuit from the PBN to the central amygdala, we used a monosynaptic rabies virus retrograde tracing method to identify neuronal projections to our genetically defined cell population of interest, which in this case would be OXTR, or oxytocin receptor expressing neurons in the central amygdala. What we find is there indeed does appear to be a cell population in the parabrachial nucleus that is projecting to our cell population in the central amygdala. We also show on the rightmost panel the locus coeruleus stained with tyrosine hydroxylase to get better context as to where these cells in the parabrachial nucleus are located. Moving forward, we then investigated the distribution of OXTR neurons in the central amygdala and PBN. So on the left, you can see this is a mouse that expresses a fluorophore under the control of the OXTR loci. And here what we find is expression of OXTR neurons in the central capsular region of the central amygdala, which is in corroboration with other studies. However, this is in another transgenic mouse line, which expresses a fluorophore directly behind the OXTR loci. And when we look in the brainstem, we find overlap between OXTR expressing neurons and CGRP expressing neurons, which to our knowledge has not been yet reported. So how does one determine the behavioral consequences of manipulating neurons in this circuit? So in order to determine the behavioral consequences of this circuit, we plan to use a direct social interaction assay, which you can see here, and is sensitive to changes in social behaviors in mice. We are also working with collaborators to develop a machine learning pipeline, which will help to quantify parameters such as social distance, which we predict might change in the presence of different bodily cues. To move our research forward, we plan to do just this. We'll first treat mice with lipopolysaccharide, the inflammatory agent I mentioned earlier, and then assess what changes occur in social behaviors. To then determine the mechanism by which these changes occur and what circuit mechanisms are contributing to these changes, we will then measure the activity of OXTR neurons in the central amygdala and parabrachial nucleus using fiber photometry. We will then also use optogenetic techniques to manipulate genetically defined cell populations in order to determine which cell populations are necessary and sufficient for changes induced by LPS. And with that, I'd like to thank my lab, my mentor, Ioana, and my lab mates, Rumi, Zara, Sheetal, Luis, and Apeksha. I'd also like to thank our funding source, the NIH, as well as our institution, Rutgers. Thank you.